Hello, my name is Megan King, Collections Manager for Rutgers Chrysler Herbarium, located in New Brunswick, New Jersey, USA. I have been working with the collections at Chrysler Herbarium for just over 4 years. I began working in our collection as an undergraduate student working towards experience-based credit. Today, I would like to tell you about how the Herbarium Army at Rutgers University adapted to a pandemic. With just over 200,000 specimens in our collection at Chrysler, 75% are made up of vascular plants, majority of which are angiosperms, leaving just under 15% being gymnosperms and pterophytes. The remaining 25% of our collection, however, is made up by other groups, as you can see here, consisting of algae, bryophytes, lichens, and fungi. As we make our way into the digital world, so do our collections. Represented here is our digitization progress. Our vascular plant collection is just over 40% imaged. This includes the pterophyte collection as well, but leaves the gymnosperms completely undigitized. The reason being is that we have yet to curate this collection to better prepare it for future digitization efforts. All other collections, however, are 100% imaged. And although many of these collections have images with skeletal data, there's a lot of work to be done as far as transcription and georeferencing. Chrysler Herbarium's home portal is the Mid-Atlantic Herbaria Consortium, also known as the MAM portal. Who makes up the Herbarium Army? Well, there are many different students participating in our collection. All students are from a broad range of disciplines, from biology, photography, ecology, plant biology, microbiology, and more, as well as in gender, sexual orientation, and ethnic backgrounds. These students are required to learn digitization work, which includes imaging, barcoding, and transcription, mountain repair, updating taxonomy, filing, and other general curations. The requirements are that they complete the number of required hours, as well as an experience-based paper and taxon report. Typically, at the end of each semester, we have a herbarium party. This past fall, 2019, many students brought in a variety of food to share with one another, from baked brie, homemade gluten-free desserts, homemade jams, and of course, Chrysler Herbarium supplied the pizza. Prospective students, current students, friends of Chrysler Herbarium, veterans of the Herbarium Army, all gathered to share stories of their experiences in the herbarium, as well as the struggles of college. Due to COVID-19, the spring 2020 party has been postponed until further notice. Since students are required to write experience-based papers based on their time in the herbarium, we have learned over the years that many of our students are gaining skills and experiences that were unexpected. During the third annual Digital Data Conference held at Yale last June, we presented a poster about the inadvertent and unexpected building of life skills from biodiversity collection digitization. Here's just a small portion of what was presented in our poster. On the left, what the students would like to get out of their experience, such as a sense of purpose and belonging, independent thinking and problem solving, learning through application, and facing the fear of making mistakes. On the right are just a few skills and new abilities that the students recorded in their experience-based papers, such as being able to work outside their comfort zones, tranquility and purpose, independent work, and time management. More times than not, it is the qualities on the right that help us achieve positions in our careers. Many students enjoy the welcoming feel of the herbarium, the delicate information-filled sheets, over the competitiveness and stress of lab work. In the herbarium, students learn more than just one task, so they challenge themselves in areas of digitization and curation to keep them engaged and feeling productive. When COVID-19 hit New Jersey, we had 16 students working in the Herbarium Army, 8 for credit, 3 paid, 3 federal work study students, and 1 volunteer. Majority of these students had only completed half of their hours, some more, some less. This was a huge change, adjustment from in-person coursework to online, then all herbarium work online as well, never mind the trauma of facing a worldwide pandemic. Thankfully, all of the students had already been trained in online transcription, which happens to be the first thing that they are shown during their initial training. So there was no rush to train those whom already have known it. After much needed training on georeferencing, we were fortunate enough to have MAM, PI Cindy Schema, and Michelle Mancini host an excellent training webinar. This training went over everything from the simple how-to the what you might come across and what is required to have in a particular data field. Chrysler Herbarium invited all students whom were already working on the portal and very familiar with transcription to take part in this training. These students were then required to spend the next week cleaning records and begin to georeference the clean records so that we would have experience with the task prior to the next Q&A webinar session hosted again by Cindy and Michelle. The bar graph on the left dictates the total number of specimens in the portal at each group of stages. On the far left, records changed out of the unprocessed stage, images that only contain skeletal data, and put into transcription stages, stage 1, pending review, or expert required. 1/3 of the records transcribed in the total were done just since the closure alone. The middle section shows records uploaded out of the total unprocessed records since the closure. These images were backlogged upon the closure, but were then processed and uploaded online. On the right, records changed out of transcription stages and put into georeferenced or completed stages. All of these records were completed just during the closure, as seen in the graph. We used GroupMe to stay connected with students during our herbarium closure. We shared pictures of furry coworkers, engaged in conversations about nature, shared experiences, as well as questions about transcription and georeferencing. We are still using this application now with our summer interns, and many of our spring interns still engage in the conversation. We think this was a great method and will continue to use this throughout future semesters to keep us all connected. Instagram posts were made about specimens they learned through digitization, curation, and research. This method of learning and sharing not only helps students to understand the importance of outreach, but allowed them to share their knowledge and experience with the broader community. Leading interns and myself each picked a day to post about something herbarium or botany related to help educate others and express the interests of our herbarium interns to those who follow us. Other herbarium students were asked to tag when posting something related to their herbarium work. While we thought we would post more, once the semester came to an end, the Instagram page got quiet. I think a more feasible option for us in upcoming semesters is to assign each student a week to develop something to be posted on the Instagram page. This would also help to engage more students than just the leading interns. There is so much going on in the herbarium, including undergraduate research. I was asked to mention that these are working titles of their projects and are subject to change in the future. Ryan Schmidt, a SEBS Rising Junior, had become particularly interested with specimens indicating ballast on their collection labels. It took little to no time for Ryan to begin digging into what the so-called ballast was, and soon turned it into a future thesis project. As summarized by Ryan, located between the cities of New York and Philadelphia, the state of New Jersey had a rich history centered around shipping and trade. During the 19th century in particular, New Jersey cities such as Camden and Jersey City were meccas of production that brought ships from all around the globe to their ports to pick up goods. In order to stabilize these ships on their journey from their ports of egress to New Jersey, merchants would place large quantities of stones, known as ballast stones, in the ship's hulls. Collected from their home ports, these ballast stones often contained large quantities of plant matter and seeds that were dumped on the New Jersey shores to later flower and reproduce. Of these many species that arrived on the shores of New Jersey, a handful of them succeeded in establishing a foothold in the state, and some have continued to thrive to this day. Cumulatively, they have had a profound impact on the state's flora, and Ryan has therefore set out to determine what species that exist in the herbaria records have had their earliest record specimens collected in places associated with ship's ballast, and whether or not they have spread beyond their counties and/or origin and are persisting in the state's flora. Through this analysis of herbarium data, he hopes to illuminate a better understanding of botanical history of the state and in turn gain a more comprehensive view of the total flora of New Jersey. Although the COVID-19 pandemic has certainly presented a large number of challenges for Ryan, he states that the previous digitization efforts by their participating herbaria of the Mid-Atlantic Herbaria Consortium have allowed him to continue his research from home. This has allowed for a strong focus on data collection and analysis that will provide a better foundation for more in-depth, in-person research once Ryan is able to return to the herbarium. Eva Popp. Eva Popp is a SEBS Rising Senior and is conducting research to understand how climate warming and urbanization affect distribution, phenology, and herbivory of two native Asclepias species in New Jersey, and the implications for eastern monarch butterfly populations. She is using the Mid-Atlantic Herbaria Consortium portal to georeference specimens and create a series of Asclepias range maps from 1850 to present day. Eva will also be examining these specimens for phenological stage and degree of monarch caterpillar herbivory, a process that must be performed in person. Because of COVID-19 research restrictions, she is unable to score specimens for herbivory or visit other herbarium to score their specimens, but has continued to georeference specimens from Chrysler Herbarium on the MAM portal. Emily Hughes, a rising junior, is working on a research project within the Chrysler Herbarium to see how trends of herbivory in herbarium specimens of white clover, Trifolium repens, have changed over time and location. She worked to score about 36 different specimens using a dissecting microscope ranging from 1865 to 2016 for 7 categories of feeding. Emily recorded each type of herbivory per cell of graph paper overlaid on the specimen. The total number of cells with herbivory can then be compared over collection date. Emily has since then been georeferencing specimens so they can be compared over geographic location and an urban gradient. Since COVID-19, Emily was no longer able to conduct her research within the Chrysler Herbarium, but fortunately, Emily has completed most of her work prior to the closure and will continue to georeference specimens. After being fortunate enough to take the position as Assistant Curator in Education and Outreach, I have found a place in all collections at Rutgers School of Environmental and Biological Sciences. Unfortunately, just 2 weeks into the position, COVID-19 hit, stopping all meetings for discussion on curation, digitization, and assessment of collections. With the list of resources provided on the SPNHC's website, collaborations with other collections, and our own history of our collections, I should in time be able to assess the collections, develop best practices, design curation protocols, as well as prepare them for a prosperous future. Once this is completed, we will be able to develop a plan to digitize them, which would then lead to more undergraduate and graduate student involvement, as well as providing a better resource for teaching and research. The SEBS collections include all of those at Chrysler Herbarium, our entomological collection, as well as many groups of teaching collections. With the support of our SEBS IT department, specifically Robert Muldowney, we will be able to build our own internal database for collections and other resources around the university. This will allow for connections in coursework and research. It seems that there is never an end to the work that can be done within natural history collections. Unfortunately, the fall 2020 semester classes are to be mostly remote, but ongoing NSF grant projects and honors projects are scheduled to continue. I would like to thank the undergraduate students and advisors for allowing me to share their work to the broader community, NSF for providing us with funding to digitize and database our collections, collaborators and aggregators for being downright awesome while constantly being there with support. A special thanks to MAM for helping Chrysler Herbarium through training, community engagement, and being a source of support through all digitization struggles.